All right. Thank you to the organizers for giving me this wonderful opportunity to come here and talk to you about one side of our work. I'm an evolutionary biologist by training, and we like to look back to Darwin and refer to his wonderful work. And I'd like to emphasize that Darwin was sort of, in some ways, the first systems biologist. So he used quite diverse types of data, right? So he looked at the fossil record. He compared geographical distributions of organisms. He looked at, he compared the anatomy of different organisms and so on. And I think if he was here today, he would be really thrilled that, much like all these other sources of data that we still know and love and use, we have one additional and very powerful source of data, which is the DNA record. And as Eric Lander says, the genome, it's a fossil record, it's a landscape, it's a whole geography of distributions. I think you all know it's not just a boring string of letters, but perhaps what you didn't know, it's a storybook that has been edited for a couple of billion years, maybe a little bit more, and you could take it to bed like 1,001 Arabian Nights and read a different story in the genome every night. I don't know about the taking it to bed part, but you can certainly take it to the lab, and that's what we do. We study the DNA record to try to understand the patterns and processes of evolution. Now, given that 1 in 2 eukaryotic genomes comes from a fungus, when I started my lab 8 years ago, one very obvious question was that we had to study fungi. So what do we study in fungi? What phenotypes do we look at? What do we try to explain? And this is the kind of fungus I like. I'm not a field biologist, so I like this. And I like this picture because it illustrates how fungi work. What's the fundamental characteristic of fungi? And that is that they live embedded in their food source. So the blue lines are the fungus, and it explores the medium. So the white cheese is basically the substrate in which it eats. And so fungi, what they do is they externally digest. So they secrete enzymes into their surrounding environment, their food, and they break it down, and then either passively or actively through transporters, bring it back into the cell and digest it. Now, this external mode of digestion poses a couple of very considerable challenges, and that is, or a major challenge, and that is that the food that particular cell, or the energy that a particular cell has spent into breaking down this food, is not just for that cell to eat, but for everybody else around in the environment. And so how do fungi solve this challenge? I believe they've solved it in 2 different ways. One is they've specialized to eating a variety of different foods, and that's reflected in their very diverse primary metabolism. And then they've developed very powerful weapons for establishing their territory and protecting their food, and that's reflected in their very diverse secondary and very potent secondary metabolism. Now, collectively, this chemodiversity, as I like to call it, has enabled fungi to basically eat almost everything, grow on any surface. I think Johan's talk was a wonderful illustration of how powerful eaters fungi can be. And I say almost everything, I would say everything, but there is one food that they can't eat, and that is McDonald's fries. So apparently they can eat diner fries, but McDonald's fries don't live there. And after Jack's talk this morning, I was thinking, I wonder what the microbiome is. McDonald's fries might be the only place where there isn't a microbiome. So they can't eat fries. Now, on the secondary metabolism side, you've already heard about aflatoxins and how powerful they are. There is penicillin. There is a wide variety of really potent toxins that fungi produce. So they're really superb. They're really fantastic chemical engineers. Now, these diversity is really critical for fungal ecology. So just to give you one very simple sort of summary example, secondary metabolic pathways tend to be very drastically upregulated when fungal pathogens infect either animals or plants, and they tend to be either downregulated or they appear as pseudogenes or completely inactivated for fungi used in fermentation. And so depending on the lifestyle of fungus, you have upregulation or downregulation or complete elimination of these pathways. So they tend to come and go. And so the way I tend to think about fungal metabolism, I consider it sort of fundamental to their feeding lifestyle, to their absorptive lifestyle. And I tend to think of primary metabolism as basically their teeth, and of secondary metabolism as basically their weapons. So much like we have, deer have, I don't have, antlers, and they use that for defense. Well, fungi, all this is happening at the chemical level, not at the level of development. And so the 2 questions I want to address today, one is how did this chemodiversity originate? and I'll be switching between primary and secondary metabolism. And then the other question I want to address is this very curious feature that fungal metabolic pathways have, and that is that imagine you have a metabolic pathway there, and you have 3 successive enzymatic reactions that convert one particular metabolite to one downstream particular metabolite, and you have 3 different enzymes. The genes encoding these enzymes, especially those that seem to be accessory, so not essential for growth. So if you delete them, the organism grows fine in the Petri dish, but ecologically has a disadvantage. These genes tend to be clustered in the genome. So they live side by side in the genome. They are independently regulated most of the times, or all the times as far as I know. And so there doesn't seem to be sort of quite a good explanation why that is the case. And so I also want to address, in the second part of my talk, why is fungal chemodiversity clustered? Why do you have this unusual feature in fungal genomes and you don't see it in the rest of the eukaryotes? So let's go to the first question. How do we generate this chemodiversity? And so a postdoc in my lab wanted to work on this question, and she sort of came up with, there are 2 major sources of gene innovation at the level of eukaryotic genomes, and one is gene duplication and the other is horizontal gene transfer. Both are really well established, happening, and very active in eukaryotic genomes. And what she did was to browse the database of genomes, and so she collected more than 200 genomes and looked at thousands and thousands of genes, hundreds of metabolic reactions from hundreds of pathways. And she looked at 3 different lineages of fungi. So I'm giving you sort of the 10,000-foot overview. So I'll collectively talk about agaricomycetes, Saccharomycotina, Pezizomycotina. There is quite a bit of diversity in the responses of these different species in each one of these bins, but I'll keep it sort of at the very high level. The number or the percentage of metabolic genes differs across genomes, and also the degree of clustering. The fraction of metabolic pathways that are in the form of clusters also seems to vary between these genomes. And so the way I'll depict the results is, or what we're interested in is how clustering sort of interacts with gene duplication and horizontal gene transfer to shape the diversity of metabolic pathways in fungi. And so I'll depict the results for each one of the 3 lineages by showing you a map of metabolic pathways. So different areas in this map represent sort of the different metabolic neighborhoods, so to speak. So you have glycan biosynthesis, and you have xenobiotics in a different neighborhood, and so on and so forth. The individual nodes in the map represent particular metabolites, and the connecting lines between the nodes represent particular chemical reactions and enzymatic reactions. And so we can attach particular genes or families of genes that are responsible for catalyzing these reactions. Now, we can go further than that and color code this map such that we can indicate whether a particular reaction or genes involved in these reactions appear to be clustered or not. We can indicate whether we see gene duplication, whether we see horizontal gene transfer, or whether we see both, and these are indicated by the different colors. And then we can show this map for the different lineages. And what's really interesting is that when you show the map for filamentous fungi, you tend to see quite a lot of clustering indicated by the thick lines, and you see quite a lot of gene duplication and horizontal gene transfer operating together. Gene duplication basically happens for almost every gene, every metabolic gene across fungi. So 90% of metabolic genes have duplicated at least once in their evolutionary history. Horizontal gene transfer happens much more sparsely. We see about 2 to 3% of genes undergoing horizontal gene transfer in their history. But what's really interesting about gene duplication happens across all these lineages. And so you'll tend to see gene duplication and again in these graphs. But horizontal gene transfer appears to be much more lineage specific. And so this is a picture from filamentous fungi. But if you go to yeast, you see a lot of clustering, so still bold lines. You see a lot of gene duplication, but you don't see as much horizontal gene transfer. And then if you go to basidiomycetes, you see lack of clustering and lack of transfer, still a lot of gene duplication. And so the different lineages seem to be sort of generating chemodiversity in different ways. Now, another way that you can generate chemodiversity is not through sort of gaining or losing genes, but through changes in the regulation, changing in where and when you activate these. This is sort of quite well established in fungi as well as in other lineages. So just to give you one example from fungi, you have Saccharomyces and you have a particular transcription factor, MIT1, that regulates a particular battery of genes. And when that transcription factor activates that battery of genes, you have a switch from yeast growth to pseudohyphal growth. Now, there is the homologue of that gene in Candida. It regulates a different set of genes. And in Candida, it's responsible for changing the morphology from white to opaque. So this is known as white-opaque switching. And in histoplasma, the same, or the ortholog of that transcription factor regulates an even different set of genes. And you get an even different phenotype. This is the yeast to mycelium transition. And so you have conservation in terms of the transcription factors. You have conservation in terms of the downstream genes. All that's changing is the wiring of how you connect that regulator to the battery of genes. And not surprisingly, this phenomenon is called regulatory rewiring. So in the context of metabolism, what we're particularly fascinated by is this observation. And that is that when we look at profiles of secondary metabolites, so we're talking about now chemicals, they appear to be species-specific. In fact, in the history of mycology, people have used presence of absence of particular metabolites as taxonomic characters. And then we know, or in the last few years, quite a lot of work has been done in sort of unearthing the diversity of regulators. And these regulators of secondary metabolism, so they're essentially controlling where and when you produce these metabolites. What's the pH out there? What's the temperature? What's the food availability, and so on, nitrogen, carbon, and so on. And these regulators are highly conserved. So you have very highly conserved regulators and very specific products. How is that mediated? So a graduate student in the lab started by asking the question of what do we know about the conservation of genes? So we know secondary metabolites are species-specific, but we don't know whether the clusters or the genes themselves are species-specific. And by looking in 4 different Aspergillus species, he was able to verify that all the clusters are basically species-specific. So when we look at these metabolic pathways, they appear to be species-specific. And the same tends to be true also for the underlying genes. So the majority, so this graph shows the percentage of species-specific genes in the 4 different species. And as a background control for secondary metabolism, which is shown in red, is primary metabolism. And you can see that we have about 50 to 60% species specificity for secondary metabolic genes and about 10% species specificity for primary metabolism. So these genes appear to be species specific. Now, how is regulation happening? We decided to look on one particular regulator. This is a protein called velvet A, that when you turn the lights off, it induces fungi to engage in sex and produce poisons. So you have sex and poison in the dark. And so we thought this is a cool transcription factor. We need to look at it. And what we did was to generate a deletion strain of velvet A, of this particular protein, and grow the fungus in that and get RNA-seq data from that, and then also grow the wild type. And we did this experiment in 2 different Aspergillus species that produce their own unique sets of secondary metabolites. And I want to show you the results first for development, because I think they'll allow you to get sort of the sense of how different secondary metabolism is. So on the left, as you look, you have Aspergillus fumigatus. On the right, Aspergillus nigilans. The proportion indicates how many genes we have. In red are the genes that are downregulated in the delta VA versus wild type experiment. In blue are the genes that are upregulated. And then in gray are the non-differentially expressed genes that are involved in development. Now I can draw lines between these 2 semicircles that indicate the orthologs, and developmental genes in these 2 fungi are nearly absolutely conserved, and so we can draw these lines. And we can also ask whether the downregulated genes in Aspergillus fumigatus are also the genes that are downregulated, their orthologs are also downregulated in Aspergillus nigilans, and I can indicate by the equivalent colors. And so you can see that we have this conservation of regulator, we have the conservation of downstream genes, but there has been rewiring. Not every downregulated gene in fumigatus, or the ortholog of every downregulated gene in fumigatus in nigilans is not always downregulated. Okay, so rewiring has happened. That's sort of the paradigm that we expect to see. Now let's look at the results for secondary metabolism. So this is secondary metabolism on the left of fumigatus, on the right of nigilans, lots of genes downregulated, lots of genes upregulated. But when I depict the orthologs, you see very few orthologs. Okay, so you don't have that many orthologs to share. And when I depict similarities of downregulated orthologs or upregulated orthologs, you really see very little. And so we believe that this is sort of an extreme version of regulatory rewiring. So in classical cases, you have conservation of the transcription factors in the two species and conservation of downstream genes and changing of the wiring, whereas here we have quite dramatic changes in the genes themselves that scrambles up completely not just the rewiring, but also which genes you're regulating. And so in some ways, the transcription factor is regulating a process, but what genes are involved in this process has been totally scrambled. OK, now, why is this chemodiversity clustered? So I want to first sort of establish and make sure everybody's convinced that these clusters are the product of natural selection. There is just no way in my mind you can generate the same genes involved in the same pathway, and some of these clusters have 20 and 25 genes, like the aflatoxin pathway, and this happening by chance. If you're not convinced by chance, I'm sure you believe that independent evolution is a telltale mark of selection. So flight has evolved from flightless ancestors multiple times. That's sort of the signature that we expect of selection. Selection is the only agent that causes directional change towards one particular type of phenotype, so to speak. We see the same thing with clusters. So when we look at different pathways across different species, we see origination of clustering independently in several different lineages. So we've uncovered examples for galactose assimilation. The 3 enzymes in the GAL pathway have independently involved in Cryptococcus yeast, in Basidiomycetes, as well as in Ascomycetes. We see this example with a 2-gene cluster involving betaine biosynthesis. You see it independently involved in fungi as a cluster, as well as in oomycetes. You see it with cyanide detoxification, and so on and so on. So selection has driven these clusters. But what is the agent of selection, so to speak? Historically, there have been 3 explanations that have been proposed. Gene clusters have originated through selfishness. They're selfish genetic elements. Gene clusters have originated because they enable co-regulation of the entire pathway, or more precise co-regulation. And then gene clusters have originated due to genetic linkage. You have favorable combinations of particular alleles at different loci, and so clustering maintains this linkage disequilibrium that enables optimal function. I have 2 things to say about these 3 explanations. One is I believe in co-regulation and genetic linkage, but not in selfishness. And I don't have the time to go through why selfishness is not a good explanation, but I'll be happy to talk to you afterwards. And the other thing I want to say is that all these 3 mechanisms are mechanisms that are genetic. They are really not phenotypes. I mean, you could get away maybe with saying co-regulation is a molecular phenotype, but the evolutionary biologist in me is resisting. So let me give you an example from a totally different set of organisms that sort of illustrates this issue. So take a look at this butterfly. This is a wonderful butterfly that its pattern is mimetic to a toxic butterfly from a totally different species. Now, there are 7 different patterns from this particular butterfly that match 7 different toxic species from entirely different families of butterflies. So this is the phenotype, right? So these 7 different patterns have evolved in response to selection, and selection is probably presumably avoidance of predation. And each one of these patterns is encoded by a supergene. So a particular location in the genome, and depending, that's in tight, absolute linkage disequilibrium, so there is 0 recombination in that supergene locus. It contains multiple genes. And depending on the allele you have at this supergene locus, you can get any of the 7 different patterns. Okay? And so in these butterflies, and supergenes are basically another word for gene cluster. And so in these butterflies, the phenotype that you have, what selection is seeing, is avoidance of predation. The genotype is selection of genetic linkage, or at the genotypic level. And then the signature of selection on the genome is that you see these supergenes and this tight, very strong linkage disequilibrium. Now, what's the equivalent of predation, right? So we have the genetic linkage down in fungi, but what's the equivalent phenotype to predation for fungi? So when you look at fungal pathways, one thing that has struck me, both for secondary metabolic pathways as well as for primary ones, is that many of the intermediates in these metabolic pathways are toxic. Perhaps the iconic pathway for that is a pathway from Aspergillus nigilans. It converts tyrosine to 2 other intermediates, fumarate and acetoacetate. And if you disrupt any of these genes in the pathway, you generate a whole variety of diseases that are known as inborn errors of metabolism. This is alkaptonuria, Garraw's firstborn error of metabolism. Pretty serious stuff, pretty serious diseases. Okay? And so a lot of these intermediates appear to be toxic. And so we came up with a hypothesis that maybe the phenotype that selection sees in fungi is avoidance of these toxic intermediates. So as making these metabolic products, these metabolites, controlling the amount of these toxic intermediates is critical. So how do we test this hypothesis? This is the test we came up with. We worked with a minimal cluster, so a 2-gene cluster, where you have a particular substrate, and enzyme 1 converts that substrate to a potentially toxic intermediate, and then the second enzyme takes that potentially toxic intermediate and converts it to the next metabolite. Okay? And so in some ways, you can think of enzyme 1 as producing the toxin or the potential toxin, and enzyme 2 being the antidote to that intermediate toxin. Now, we have the genes encoding these enzymes in 2 different conformations in the genome. So they can be dispersed in different parts of the genome, unclustered, or they can be side by side. So in these cases, we call them double neighbors because they're not only neighbors in metabolic space, but also neighbors on the genome. And our prediction would be that the unclustered pathways, these unclustered enzymes, would handle intermediates that have much smaller degree of toxicity than the clustered ones. Okay? So we can go and identify, looking through KEGG maps and other metabolic databases, enzymes that are double neighbors, and we can look in fungal genomes and see are they clustered or no clustered in the genome, and then we can take these intermediates and scan biochemical databases that report toxicity of some kind or other for these clusters. And we can measure toxicity in 2 different ways. One is by LD50, so the amount of dose you need to kill a certain, 50% of the population of cells or mice or whatever you're looking at, or by looking at how reactive the compounds are. So are they like hand grenades, or do they have very precise modes of chemical action? And so in both of these cases, when we look at LD50, we see that pairs of genes, double neighbors, that are clustered in divergent orientation, so they look away from each other and likely have a shared regulatory element, appear to be much more handling much more toxic intermediates than clusters in either of the 2 orientations or unclustered species. The line that indicates the null expectation is the black line in the middle of the graph. And the same seems to be happening also with reactive compounds. But in this case, we see toxicity associated with clustering, not only for divergently oriented clusters, but also for collinear clusters. And so these are the results that seem to support it. Let me give you just one example of the many clusters that we have, or the many pairs of genes that we see clustered, and that we clearly see a toxic intermediate in the middle. This is a 2-gene cluster that the first gene, choline dehydrogenase, produces a toxic intermediate called betainaldehyde. It's so toxic that plant biologists use it occasionally as a selectable marker for transformation. And then you have this gene betainaldehyde dehydrogenase that rescues basically the toxicity. You see this gene cluster in quite a few fungal species, and this is one of the clusters that has independently evolved in oomycetes. Now, I want to close by addressing one question that I often get at the end of seminars, and I was getting tired of not having a good answer, and so we thought to do a little project out of it. And that is that for some of these pathways, and here I'm talking about exclusively primary metabolic pathways and not secondary metabolic pathways, you also see them in other organisms. I just showed you a few slides ago the pathway for tyrosine catabolism. And I mentioned that there are a bunch of human genetic diseases that are associated with that, so that indicates that that pathway also exists in humans. But in humans, that pathway actually was discovered by looking or studying Aspergillus. We found the human genes through study of Aspergillus, not me, Miguel Penalva's group. But in fungi, you see the pathway clustered, and in humans, you see the pathway dispersed in 4 different chromosomes. So chromosome 12, chromosome 3, chromosome 14, and so on. The toxicity is still the same because we know there are human diseases associated with disruption or mutations in these genes. So why fungi have the clusters and why humans have exactly the same pathway and it's not clustered. There are many reasons that I don't want to go into. There are huge differences in population size that affect the efficacy of selection. Fungi are much better able to take advantage of selection than the human population or mammalian populations, which are smaller in size. There are significant differences in reproductive lifestyle. Fungi can engage in sex as well as asexual reproduction that tends to maintain this clustering. But the point I want to focus on, and I find particularly interesting, is that in fungi, if you have a toxic intermediate, as it grows on the Petri dish or as the mycelium grows, if you have a toxic intermediate and it has a disadvantage in growth or it has a lethal phenotype, the whole colony disappears. In mammals, if you have a toxic intermediate, what happens is you destroy your liver and your kidney over time, but not much else is happening, at least in the beginning. So in some ways, mammals or organisms, animals with morphology and three-dimensional features, in some ways have compartmentalized dangerous chemistry to particular tissues. And so we came up with this hypothesis that maybe, in fungi, it pays to sort of restrict toxicity through the clustering, through favoring clustering of genes. And maybe in mammals, it pays to restrict toxicity to a different mechanism. And that mechanism may be that making sure that your genes that are involved in that pathway are expressed in a specific tissue. So tissue-specific co-expression would be the signature for humans, and clustering would be the signature for fungi. So this is the hypothesis that the clustering of genes in fungi and the tissue-specific co-expression of their orthologs in humans are basically analogous responses to the same selective pressure, avoiding getting killed by these toxic intermediates. So how do we test this? We can test this by taking two genes that are clustered in a fungus, gene A and B, and looking for their orthologs in humans. These orthologs all the time, in all the examples that we've seen, are unclustered. And so we have human gene A and human gene B that are orthologs to the fungal genes. And then we can measure their expressions in humans. And there are many very nice data sets that now do so. And so you can look at different tissues, how these genes are co-expressed. And then we can measure the correlation of that human gene pair. So how is human gene A correlate in its expression with human gene B? And the different letters correspond to the levels of expression of that particular gene in the 6 different tissues that we surveyed. We can also do that as a control for genes that are not clustered in fungi, and find the non-flow pairs, so the pairs that are not linked in fungi, and do exactly the same experiment. So we have case and control. And the expectation is that for cases where we have gene pairs that are linked in fungi, we would expect to see very high tissue-specific correlation of their human counterparts. And in cases where we're dealing with non-toxic pathways, we wouldn't see clustering in fungi, and we wouldn't see strong tissue-specific co-expression, or as strong tissue-specific co-expression in humans. It's stats at this point. And so what we're finding actually is the case. So we find support for this hypothesis. So we see that these fungal-linked orthologs, so human genes whose orthologs are linked in fungi, tend to be significantly more co-expressed in humans. So this is the solid black line. That's the flows. And the thin line is the non-flows. There is tissue-specific co-expression and correlation because we're looking at genes in the same pathway. But if you're linked in fungi, the tissue-specific co-expression that you observe in humans is way over and above what you would expect just from participation in the same metabolic pathway. And we not only see this in humans, but we see it in chimps and other primates. And so what I'm trying to tell you is something that still staggers me when I keep thinking about it. And so what I'm really telling you is that you have 2 genes that are sitting side by side in a fungal genome, and that participation, that location in the fungal genome basically tells you something about how these 2 genes are going to behave in the human genome. I find that astounding. I have no explanation other than saying that evolution is really powerful. And we have signatures of selection that can be really informative, I think, as we think about the human genome and sort of very practical applications to curing disease and so on. and that information encoded in genomes is a very rich resource that I think we're only beginning to tap. So I'll stop there and thank the people that did the work. This work on clustering originated when a very talented postdoc who now has his own lab at Ohio State, Jason Slott, came to my lab as a first-year researcher. That was a gift from heaven. And I've been blessed to have more great people coming in. So the work on the origins of fungal chemodiversity has been led by Jen Weiscaver, a postdoc in the lab, and Abigail Lind, a graduate student in the lab. And then the work on the toxic intermediates has been led by Chris McGarry, a postdoc in the lab, and more recently by the help of Haley Idem, a graduate student in the lab. So I'll stop there and take any questions. Thank you for your time.
All right, we're off schedule, but maybe one really quick question.
I have a question about the production of the secondary, the intermediate. You said that they're toxic, but why are they beneficial to fungi if they're toxic in the first place?
So they're beneficial because they're exported, usually. So most secondary metabolites are made in particular compartments inside the cell, and they're exported extracellular, and so essentially they're fumigating the area. In many secondary metabolic pathways, and this is sort of active research, but more and more we're seeing genes inside the clusters that confer resistance to that cluster. And so if you take a particular gene and give it to another genome, you put it in yeast, suddenly yeast gets resistance to gliotoxin, for example. And so they're making hand grenades, and they're just throwing them outside the cell against everybody else. But they have some protection against them.
All right, thanks so much, Antonis.
Thank you.